Hello, everyone, and welcome to our 18th lecture in our course on brain and behavior. And this is a lecture, no new material, it's just a review for exam two. But hopefully this helps you prepare for exam two in terms of what information will be most focused on, but especially how to organize your notes, how to organize the content, so you can think of the material for exam two in a helpful way, as not just a sort of list of facts to be memorized, but as a more cohesive whole. And so we'll talk about cause and effect relationships, how different content in these lectures relate to one another. So we'll go over that. We'll sort of organize the content, focus on particular areas, and again, that should get you ready for exam two. In addition to using things like your notes, obviously, practice quizzes, going back to class exercises, that sort of thing. Okay, so the structure will be what you've already seen before. There will be multiple choice questions. That's around 15% of the exam. As always, you don't need a Scantron. You'll just work right on the exam itself. There are going to be fill-in-the-blank questions and matching questions. The fill-in-the-blanks, you will have a word bank. And of course, matching questions, you have the possible answers on one side and the questions or terms on the other, and you just match them up. And when it comes to, especially the matching questions, some of those questions are going to be about cause and effect, about sequences of events. And so it's not always just matching up terms with definitions. We know that cause and effect is an important part of this class. Understanding things like the processing of sensory information as a series of steps, or the effects that drugs have on neurons or the body or the brain or behavior, a sequence of steps, how research methods work. So when you're studying, and especially getting ready for those matching questions, think of those procedures and those events that happen step by step exactly as that, not as a list of definitions or ideas to be memorized, but as a logical chain of events. As always, short answer questions. So you'll have to get comfortable putting things in your own words, explaining concepts, describing results. There'll be diagram questions again. And as I've already mentioned, the diagrams that are in these slides are the best candidates for those diagram questions. It could be explaining the content of a diagram. It could be labeling things, so removing the labels that exist on a diagram and you have to fill them in. So being familiar with the class content, especially that content that is best demonstrated visually, that's hard to express verbally, or at least takes a long time. So things like structures, concepts that require graphs, these are always good candidates for diagram questions. So keep that in mind. And as always, there's our essay. In this case, it's the virtual lab exercise that you did. So be able to explain what you did, what the results were, what they mean in your own terms. That's always going to be part of the exam. So that is the virtual lab. Hopefully you took notes, and we discussed it in class. So keep that in mind as you prepare. And what this exam covers is what we've covered since exam one. So our unit on the senses, our unit on research methods, and our unit on psychoactive drugs. And those consisted of different numbers of lectures, and that's going to be reflected in the exam. The more time we spend on a topic, the more it will be covered in the exam. So for the senses, understand those sensory systems in general, what sensory transduction is, both what it means literally, what that word means, and how it works for each of the senses. So the senses each work differently. So know what each sense detects, whether it's chemicals, whether it's photons, whether it's mechanical pressure. And again, cause and effect relationships, what that process is for each of the senses. How do we get from, so this is a diagram from your book about processing sound, how do we get from sound waves to neural impulses? For the visual system, how do we get from photons to neural signals? So be able to explain those steps for each of the senses. This is the diagram for your somatosensory sense. How do you get from mechanical pressure, stretching of the skin, to action potentials? Well, in the case of your somatosensory system, those channels are physically pulled open when the membranes are stretched. And when those channels are pulled open, sodium flows in, you depolarize the membrane, and then you get an action potential, you get neurotransmitter release, and then those neurons convey action potentials all the way to the brain. So know each of those steps for each of those senses. Also, the basic plan for getting information to the brain. So we already know that the thalamus is very important, and we know that the thalamus is involved in all of our senses, except for our sense of smell, olfaction. So understand that basic plan, where cortices are for each of the senses. So more specifically, for the somatosensory sense, know the cells that are involved. You don't have to go into details as to which are fast adapting, which are slow adapting, but know their names, be able to recognize them. Say a word about what each one detects. The auditory system. Know the structure of the ear. We spent a bit more time on the auditory system, not as much as on the visual system, but about the second most focused on sense was audition. So know the structure of the ear, the outer ear, middle ear, inner ear, parts of those various zones. Understand hair cells. How do they work? How do you get from movement of the basilar membrane to neurotransmitter release? How are they built structurally? So they're different from run-of-the-mill neurons. So be able to describe and understand what hair cells are and how they work. We talk about how we localize sound, and that's important as well. So we can tell the difference between a sound coming from the left or the right based on two phenomena. The relative loudness for each ear, and the relative timing for each ear. So if it's coming from the right, then that sound will be louder to the right ear, and it will arrive at the right ear first. And sound waves are slow enough that we can detect that timing difference. For localizing sounds in terms of elevation up and down, we know that the pinna, the outer ear, differentially filters certain frequencies of sound, and so a sound that is coming from high up will sound a little bit different in terms of frequencies than a sound that is coming from ground level or below us. So those are three ways that we can tell where a sound is coming from. The forms of deafness. So we have conduction, sensory neural, and central deafness. Know what those terms mean and be able to recognize the various forms of deafness. The gustatory system, our sense of taste. Know the structures of the tongue. There aren't that many. But know the difference between a papilla, a taste bud, a gustatory receptor cell. So those are differences in scale. And they have sort of a hierarchical structure. Taste buds are composed of gustatory receptor cells. A papilla is covered in taste buds. So know that hierarchy. It's pretty simple, but know it. The olfactory system, we spend a little more time on than gustation. So understand how this works. Understand the cells that are involved. So we have olfactory receptor neurons. We have receptors on those neurons, each receptor corresponding to a different odorant. So those receptors, as we discussed, and this diagram illustrates that, are metabotropic receptors. So we have these receptors that bind to various odorants, and that's how the sensory transduction process works. An odorant binds to the receptor. That receptor changes its shape. It sends chemical messengers that open up ion channels. And so that's how we get from the presence of an odorant to the release of neurotransmitter. The vomeronasal system is sort of a fun one, and we talked about it a little bit, and know what it's for. So the vomeronasal systems detect pheromones. And that's especially useful for other mammals and other animals. Humans, of course, we don't have one, or at least ours may be vestigial. It doesn't work. It may be present in a very small sense. But know what it's for, and know that we don't have one. The visual system. So we spent an entire lecture on the visual system. It was the sense that we went into most detail for. So spend proportionally more time on it when you're studying. So understand what photoreceptors are. Know how they work. Again, sensory transduction. How do we get from a photon to action potentials, to neurotransmitter release? Understand the structure of the retina, especially as it relates to those photoreceptors. So we have more cones in the fovea. That's why we're most sensitive to color differences. But we also need to have fairly high light levels to use the fovea, because cones need more light than rods do. So rods are more common out in your peripheral vision. They're not as sensitive to color. In fact, they only detect things in shades of gray. But they are better in low light levels. So understand how that difference relates to where rods and cones are concentrated in the fovea. Also, when it comes to retinal structure, understand that photoreceptors, you don't need to know all the little kinds of neurons that are in between the photoreceptors and the brain, but understand those photoreceptors, understand the ganglion cells. So they collect information, their axons form the optic nerve, and then pass out of the eye. And that has consequences for how information is processed. That's part of our steps of cause and effect. We have light coming in, processing in the photoreceptors. Remember, those photoreceptors actually hyperpolarize when light comes in, so they stop releasing neurotransmitter. But beyond that, the retinal ganglion cells collect signals from other cells in the retina. Their axons bunch together into the optic nerve, and those signals get sent to the brain via that optic nerve. And that optic nerve being there and exiting the eye has consequences. So the structure of the optic nerve, it's so big that you have that blind spot. So understand what that means for retinal structure. Understand why that's important for those retinal ganglion cells. And then in terms of processing, we also have the ability to perceive colors. And that's the function of our cone cells. So different wavelengths of light lead to different activity levels in the different cone cells because they're each sensitive to a different wavelength of light. We have 3 different wavelengths that cone cells are maximally sensitive to. So understand how that leads to color perception. We also have the phenomena of agnosia, which is not so much at the level of the retina, it's in the brain, but it's one of our information processing steps. So we have to, much later on in the stages of visual processing, we have to be able to recognize objects. And not everyone can do that. And there are different forms of agnosia. Don't worry about the individual forms of agnosia so much as what agnosia is and how it relates to the brain. So keep that in mind. But again, vision we covered more heavily, so study it more heavily. Research methods. So we spent a whole lecture on research methods. Know what each technique measures. You don't have to write a paragraph about it, but in a few words, be able to say what each technique does. So EEG, electroencephalography, DTI, diffusion tensor imaging, PET, positron emission tomography, TMS, transcranial magnetic stimulation, CT scans, or CAT scans as they're also known, fMRI, and that F is important, functional magnetic resonance imaging, as opposed to typical MRI, magnetic resonance imaging. Those are different techniques, even though they use the same machine. Electrophysiology is another one that we talked about, and it's quite different from imaging techniques, for example, or other large-scale techniques. Electrophysiology, very detailed. Lesion techniques. So lesion techniques are destroying part of the brain, not something we do on purpose in humans, but it has its place among research methods, either doing it intentionally in animals or studying patients that are human that have had accidental lesions. Electrical stimulation we've talked about a little bit in our research methods lecture, but throughout the course we've talked about it. How do we create a sensation or perception in the brain by stimulating it electrically? How do we cause someone to move their body by stimulating it electrically? We also mentioned in this lecture how electrical stimulation, in the form of deep brain stimulation, can resolve or at least treat the symptoms of various disorders. So for each of those methods, be able to, again, describe what each does in a few words. But we talked about the difference between structural techniques and functional techniques. So understand that distinction between structure and function as it pertains to research methods. And for each of the techniques above, be able to say which one it belongs to. Does the technique work at a structural level, how the brain is built or how neurons are put together, or the functional level, which neurons are active, which parts of the brain are active? And so a technique might manipulate, or it might observe. It might change something about the brain, or it might simply passively record something about the brain. And this interacts with structure and function because you can manipulate brain structure with something like lesion, destroy part of the brain, or you can manipulate brain function, neural activity, with something like TMS. TMS doesn't hurt the brain structurally, it doesn't change how it's built, but it changes which neurons are active. So be able to classify each of those techniques based on structure, function, based on observation or manipulation, and also based on whether it's invasive or non-invasive. Does it involve breaking the skin, going through the skull, disrupting brain tissue? Obviously, a lesion is very invasive. It's destroying part of the brain. Electrophysiology is invasive. You have to put an electrode in the brain, and that requires, at the very least, opening up the skull. Something like EEG or fMRI is non-invasive. You don't have to break the skin. You can measure these things either on the scalp or by putting someone in an MRI machine. It doesn't involve breaking the skin, opening up the skull, doesn't destroy tissue. So those techniques are non-invasive. And of course, techniques that are observational and look at function can do that a number of ways. So techniques like fMRI measure metabolic activity in the form of blood flow. When an area of the brain is more active, it needs more sugar, it needs more oxygen, it needs more blood. So blood goes to that area, and that's what fMRI is measuring. But that's just one way of detecting neural activity. You can also detect neural activity directly through electrical measurements like EEG or electrophysiology. So these measure action potentials or averages of action potentials by using electrodes, detecting changes in potential. And of course, there's chemical activity. We saw that in our virtual lab exercise. So there we're measuring the presence of dopamine. And so that is a way of looking at neurotransmitter release. We're not measuring action potentials in terms of electricity. We're not measuring metabolic activity. We're measuring the presence of certain chemicals. So that's another way of looking at neural activity. And also, we talked about how techniques differ in terms of their spatial and their temporal resolution. So for each of the techniques above, think about where they lie on those two spectra. What is the spatial resolution of each technique? What is the temporal resolution of each technique? So EEG, very fine temporal resolution. Can take thousands of measurements a second. Relatively poor spatial resolution. You're only measuring billions of neurons at a time. You have an area the size of a silver dollar, as opposed to something like electrophysiology, which also has very fine temporal resolution, but also very fine spatial resolution. You're measuring a single neuron. You can only measure one at once, which is why you may not want to use electrophysiology, but you have a lot of detail about that neuron. And that's in contrast to something like fMRI, still measuring billions of neurons at a time, a little bit more fine resolution than EEG perhaps, but roughly similar, billions of neurons. Temporal resolution, not as great. You're taking a measurement every couple of seconds. Something like a lesion lasts for years. and also has a very coarse spatial resolution. A lesion could be millimeters in diameter. It could be the size of a silver dollar. It could be bigger. And so think about each of those techniques and how they differ in terms of spatial and temporal resolution. For our unit on psychoactive drugs, know the difference between an agonist and an antagonist. And this is a big deal. This is an important part of understanding how drugs work. It's one of the most critical ways of defining a psychoactive drug. We had a class exercise on whether drugs were agonists or antagonists. I guarantee this will be part of the exam. Also understand the difference between direct and indirect agonists and antagonists. So an agonist activates the receptors for a particular neurotransmitter. In a sense, it helps the neurotransmitter do its job. It might imitate the neurotransmitter and activate those receptors because the receptor thinks it's that neurotransmitter. It might increase levels of the neurotransmitter. It might increase how much neurotransmitter is released. So you could produce more, you could release more, you could prevent reuptake. So keeping neurotransmitter out in the cleft. All of those are ways that agonists can have their effect. Antagonists, you can prevent production of a neurotransmitter. That's obviously going to decrease levels of that neurotransmitter, going to keep those receptors from being activated. You can prevent it from being released. You can block it at the receptor level. You could structurally imitate the neurotransmitter, but when the drug binds to the receptor, it keeps it from being active. It blocks it, keeps it closed. So that's an antagonist. And remember that direct agonists and antagonists affect the receptor directly. They bind to the receptor. Indirect ones is sort of covers everything else. It doesn't bind to the receptor, but it somehow affects the neurotransmitter system in some other way, whether it's amount of neurotransmitter produced, amount released, how reuptake works, how it's broken down. Those are all indirect ways for drugs to work. And remember the steps of synaptic transmission. So this is going to refer back to earlier content. Not at the same level of detail as we covered before, but know how each drug affects synaptic transmission. We'll go over the individual drugs here in a moment that you need to worry about. But remember that those steps of synaptic transmission relate to whether it's an agonist or an antagonist, whether it's direct or indirect. And you have both presynaptic and postsynaptic ways of affecting synaptic transmission. The postsynaptic ones are almost all dealing with the receptors themselves. They're direct. The presynaptic, there's a lot more variety. So keep that in mind. It can have presynaptic or postsynaptic effects. And be able to identify specific drugs. So it's described what the drug does, what neurotransmitter system it works on, or what category a specific drug belongs to. Be able to do all of that. So if you're given a category of drug, be able to name a specific member of that category. If you're given a drug itself, a specific drug, be able to name what category it belongs to. And be able to compare and contrast the various kinds of drugs that we talked about. So one way that we can distinguish between different kinds of drugs is using that ED50 and that LD50 metric, the effective dose and the lethal dose. So know what those terms mean. I mean, know what they mean literally in terms of what those acronyms stand for, but also what it means in terms of drug safety, drug effectiveness. So when it comes to drug safety, we know that we want the ED50 and the LD50 to be far apart, because that gives you a lot of wiggle room. You can deliver enough of the drug to be effective without having to worry about it being lethal. Whereas if the ED50 and the LD50 are very close together, well, now there's very little margin for error. So understand how that works and what the ED50 and the LD50 mean for drug safety. The process of addiction. Know what addiction means, what dependence means. Know what neurotransmitter is most commonly involved in highly addictive drugs. So certain classes of drugs are more addictive than others. We know that, for example, the stimulants are at higher risk for dependence and addiction than, for example, the hallucinogens. So understand why, because of the differences in neurotransmitters that are involved. The process of tolerance. Know what tolerance is. know how it relates to changes in the brain, know what it means for behavior. So you have to take more of the drug in order to get the same effect because of tolerance. So understand how that process works. So when it comes to the categories of drugs that we talked about, be able to compare and contrast them. What's the difference between a stimulant, a depressant, an opioid? And we had some subcategories for these. So we had the hypnotics, the anxiolytics, the dissociatives. So be able to name an example of each category, know what they are used for, what these labels mean. So what are they used for in a legitimate sense? Why would a doctor prescribe an anxiolytic? Why would a doctor use a dissociative during surgery? But there are also illegitimate uses, recreational uses. So know why people use these different substances, even recreationally. People use alcohol to relax themselves or to help themselves sleep, even though no doctor would prescribe alcohol. Why would a person use a stimulant? It could be to keep themselves awake. It could be in order to get the euphoric effects. If someone's in chronic pain, even if they don't have a prescription for an opioid, they might still use one recreationally. So understand that connection between the effects of a drug and its use. And when it comes to knowing the individual drugs, focus on the ones from lecture. So we talked about nicotine, which is a stimulant a fair amount. Understand how nicotine works on neurons. Understand what its effects are. Other stimulants, cocaine, methamphetamine, know what neurotransmitters they affect and how they affect those neurotransmitters. Know what the behavioral effects of these drugs are, and also the dangers. So cocaine is toxic to tissue. Methamphetamine kills dopamine neurons. So know those effects. Alcohol is a depressant. So again, people might use it to relax themselves or to put themselves to sleep, and understand how that connects to neurotransmitter systems. What neurotransmitter do depressants affect? Marijuana is its own category, so it's both a category and a specific drug. So know how it works. How does it affect neurons? Through what receptor? What are its behavioral effects? The psychotherapeutic drugs, we focused on antidepressants, and in particular the SSRIs, the current generation of antidepressant. So know how SSRIs work. What step of synaptic transmission? What neurotransmitter? So in other words, know what SSRI stands for. We didn't really focus on a specific opioid. We talked about some history, what receptors they affect. Heroin is a fine example, so if you study heroin, that'll be good. Know how opioids work, what effects they have behaviorally, what effects they have on neurons. The hallucinogens, we talked more about MDMA, a little bit more about LSD. So know that these are hallucinogens. Know what neurotransmitter they affect. Know the relative risks. So LSD, low risk of overdose and dependence. MDMA carries more risks. Know what they are. And again, referring to the content above, the dissociatives count as hallucinogens. So be able to name some dissociatives. Know how they work. And then finally, the virtual lab experiment that you completed. So know what the purpose of the study is. What is it that you're looking at? What neurotransmitter did you study? What drug did you study? What was the method? What did you do? So be able to describe that experiment in a paragraph in your own words. What were the results? What did you observe? So you spent time in that experiment making observations. What did you find out? What did you see? And then what do those findings mean? How do those relate to concepts like drug consumption, drug seeking? What does that tell us about human behavior and things like drug dependence in humans? So know the details, but also know the generalities, know the implications, know what this means for drug use in general, what it means for the interaction between neurotransmitters and drug use in general, know what it means for humans and human behavior and our understanding of processes like addiction. Okay, that should get you ready for our exam. So if you have any questions, obviously feel free to ask me prior to the exam. And go back, use these slides, use this video to help you focus as you study. Use those practice quizzes. Use class exercises. Obviously go back, look at the book, and study your notes and the lecture slides. So if you do all of that and focus on the topics we've covered here, you should be in pretty good shape for that exam. So with that, good luck on the exam, and I will see you in class.